opportunity to thank Gil Omenn once again for everything he's done for us over the years. I think folks know that Gil was brought in here now 20 years ago to be our first Executive Vice President for Medical Affairs, a new position that was created. We were so lucky to have him. That brought the hospitals and health centers together with the medical school, which is a theme under Michigan Medicine that we're still doing today. And after he stepped down from that position, the question is, what was he going to do next? Well, I think he went back to what he really loved, which was proteins, the proteome, and mentoring and leadership. And the theme of this whole Omenn lecture in Gil's involvement with the university and with us is about leadership and mentorship, really, when you boil it down. And he joined with me and the two of us together, and we're really honored to have David States here, our first graduate program director for the Bioinformatics graduate program. Welcome, David. It's great to see you. In 2005, the program was started. David, he came later. Phil Andrews and Brian Moldover and Dan Burns, Mike Savageau helped to conceptualize and pull together the idea of the Bioinformatics graduate program in 2009. And you know, it started to crawl around. And then in 2005, Gil and I started the Center for Computational Medicine and Bioinformatics, which ran all the way to the end of 2011. And during that time, we got a lot of things going and really started the whole scale-up of computational medicine, computational biology, bioinformatics here at the university and in the medical school, and led to the creation of our department. You know, we got out on the national stage with our NCIBI, one of the NCBCs, and got to help really get the whole CTSA going here, which is now in year 13. And, you know, Gil really did start his long and continuing leadership of the HUPO, the Human Proteomics Organization, and other organizations. And then we were so lucky in 2012 to start up in January 1 and to have the chance to build a department as part of the medical school and the basic sciences. And that's really gone well, you know. We have now 13 faculty, soon to be 16. And actually, I was telling David that Arvind Rao was going to start in a couple of weeks, and then he just popped up. He's here. Where are you, Arvind? There he is. He's up there. And that's really neat because Arvind was one of our first graduates. So he's come back after a successful postdoc at Carnegie Mellon in a faculty run at MD Anderson. He's back home. It's wonderful. And he'll also be a research professor in MIDAS, our Data Science Institute. We've got a couple more recruits coming, so we'll be up to 15 or 16 faculty on the instructional track and 8 research track faculty at all levels. Many that are associated with our bioinformatics core, you know, and then 14 affiliates. So we've got an up and going department, and it will continue to grow. So it's really exciting. And it's great to see how many students and trainees and postdocs are engaged with all this. And I called down to the office and found out that we've now graduated 81 PhDs from the program, David, and over 50 master's students. And what's neat is that so many of our PIP students are joining up and getting an embedded master's in bioinformatics. I expect that we're going to see a majority of the PIP students select that, just as our students, many of whom get master's degrees in statistics and biostatistics. June joined us as associate chair for research in 2016, and Margie, who will be back from Shenzhen soon. She sends me at least 2 emails a day, and she's co-chairing a data science conference for the next 2 days in Shenzhen at the Chinese University of Hong Kong. Shenzhen. Now, in terms of the Omenn lectures, Atul, who Gil will introduce in a minute, is our 5th lecturer. It started in the series. We started off and we raised $100,000, actually about $120,000 into a fund so that we could have an Omenn lectureship ongoing that would involve allowing us to recruit the best leaders, around that we could find and inspire us. And so far, you can tell that we've done really well with that list. And, we're picky. So let me put it that way. Started off with Lee Hood. And then there's a little secret. Atul's mentor, Zach Kahane, came in 2014. David E. Shaw, who, he mentored a lot of us into thinking about how we can use our skills for other things in 2015. George Post last year. And then, there isn't, it's now into 2018, but this is the 5th. It's in the academic year. And so we're just delighted to have you, Atul. More about that in a minute. We'll let Gil do that honor. Mentors. Here's Dr. Omenn's major mentors. And, this is really the key. It's all about mentorship. And the connection on mentorship is really, Gil's my mentor. Gil has mentored Zach. Zach was Atul's mentor. It's like a network. These are Gil's mentors, starting with his parents. This fellow here was the 4th leader, the president of Israel in the mid-70s. He was also a biophysicist. And I think that Gil was kind of a closet biophysicist. Kristen Antonsen was a biophysicist in a lot of ways, biochemist, protein structural biologist. And so the roots for structural biology and proteins go back into the 60s with Gil after he got out of Harvard Medical School. And of course, he's been involved in government and policy. It's a picture with Frank Press, President Carter, when he worked at the Office of Science and Technology Policy as the Associate Director. And so everybody has mentors. And Gil has been a phenomenal mentor to all of us. And this is really what this is about. Celebrating that and learning about leadership by looking at the example of leadership. And that's what the theme is. Here's a picture. I couldn't find one with Dean Willistoff putting up on the stage. But this is the first Omenn lecture over across the street. And I'm going to have a chance to bring Atul over to see the Omenn atrium where this is. We usually like to hold the lecture there. We have the reception there. We weren't able to do it this year, but at least we're able to have it in our home turf here at Palmer Commons. And, there's Lee's title and talk there. What I remember talking to Lee a couple years later, and he still talked about, I didn't get any pictures, at least weren't able to find that incredible student lunch we had down in 2036. He absolutely loved that. And, I know that myself, having been the beneficiary of Lee Hood's mentorship over the years, back in the early days when we set up the first computational systems. He used to spend so much time here at Michigan with us so generously. This is Zach and I looking at Gil and talking back and forth when Zach came around. And, I've got more pictures that I think we're going to pull together and share. But it's my opportunity as a mentee of Gil Omenn's to hand off the microphone to our leader, our colleague, our friend, and have him introduce the 5th Omenn lecture. Gil, it's my great honor and pleasure.
Okay, thank you very much, Brian, and thank you all for coming. It's a special opportunity here at Atul Butte, here on home turf. I've heard him many other times, and it's part of our special leverage to bring exciting people here for everybody else to benefit. He said that he was amazed himself that he'd never been here all these years, so it's a signal day for you and for us both. Atul, as you heard a little bit, I'll give you a little more background, is from Long Island, went to Brown University, majored in computer science. He then worked at both Apple and Microsoft as a software engineer. Gives me a lot broader sense of what's potentially feasible. Then he did the MD at Brown, trained in pediatrics and pediatric endocrinology, and was mentored by Zach Kahane at Boston Children's Hospital and Medical Center. And meanwhile earned his PhD from Harvard Medical School and MIT under their joint Health Sciences and Technology program, which is always thought of with regard to medical students, but in this case, per resident and fellow, which is good flexibility. And I'm proud to say that Harvard, according to its recent dean, announced in 2015 that they were going to learn from the University of Michigan model of a center growing into an academic department and created the Department of Biomedical Informatics at Harvard. So we're connected. And of course, you heard Zach was a speaker here 3 years ago. Atul has had a special role in the development of translational bioinformatics, clinical bioinformatics. In fact, one of his questions was, how well are we connected with that side of the operation here, which is still on our to-do list. But I first met him when he was the organizer of the Translational Bioinformatics Summit in San Francisco, which has become a now 11th year annual event in San Francisco under AMIA, the American Medical Informatics Association, where he's a big leader. He holds the wonderful title of Priscilla Chan Mark Zuckerberg Distinguished Professor, as well as inaugural director of the Institute for Computational Health Sciences at UCSF. Now, this was an earthquake in California when Atul, who was thought to be locked into Stanford for posterity, picked up and moved to the institution to the north in San Francisco. And of course, he's a man of great vision and negotiating skill. So it was not just San Francisco. It's all of the UC medical campuses put together into a 6-institute combined data system and precision medicine, medical care quality improvement initiative that will benefit many millions of people in California and globally. I think we're going to hear about that a bit today. So here's the title, Translating 1 Trillion Points of Data into Diagnostics, Therapies, and New Insights in Health and Disease, Clinical, Translational, and Basic, all in one. Welcome, Atul.
Thank you.
Great.
All right, great. Well, thanks for having me here. It's a real honor, especially seeing the other 4 names up there. It's a real honor to be considered the 5th on the list here. And thanks to all of you for coming out in the afternoon here. I'm sorry I messed up your numbering of the years. It took forever for us to finally schedule this date. So but we made it, and we're still in the academic year, so that's all good. So first of all, I'm going to talk about a lot of different things, but I'm a medical doctor, so I usually have to start with my conflicts of interest. So I have just a few. Suffice it to say, I started a couple companies. I consult for a bunch of companies. You might not want to believe another word I say for the next hour. I wouldn't blame you. But I'm most proud of the bottom right. Those are all the companies started by my graduate students. More than half my graduate students now start companies, even if they go into academia. And they do this with the most amazing platform in the world. It's simply data. It's often big data. It's often open big data. I'm going to show you how they do it. I'm going to show you how I do it. And maybe I'll convince you this is the most amazing time to be in biomedical innovation and entrepreneurship. Now, I'm spending a little bit of time on this slide for a reason, right? In my lab, we do have a saying that if you want to change the world, you can't just keep writing papers about it. If you've actually built a tool, you should use that tool. If you've used that tool and made a discovery, and especially could help patients or families or physicians, you should take it forward. Maybe file some intellectual property. If no one licenses that intellectual property, it's up to you to start the company now. No one else is going to do it. So a lot of physicians use these kinds of slides as a slide of shame. This is my slide of significance. This is how we're taking beyond the lab. So if you haven't heard, we're in the middle of this data deluge. A lot of covers of a lot of magazines now. This is my favorite one from The Economist magazine about 5 years ago. This guy's collecting ones and zeros with the umbrella, And this is the issue where they announced that the human species generates 2 zettabytes of data every year, 2 zettabytes. If you forget your metric prefixes, it goes kilo, mega, giga, tera, petabyte, exabyte, zettabyte. Of course, the next year it's 4 zettabytes and 8 zettabytes and 16 zettabytes of data. I'll be the first to admit most of these zettabytes of data are YouTube videos of kittens playing with pianos and yarn, Entertainment value, but no scientific value, perhaps. But there are scientific data sets in the zettabytes. So, for example, the Large Hadron Collider in Europe generated petabytes of data. By the way, all that's open data. Petabytes of data for those scientists to find new subatomic particles, right? But my favorite example now is NASA. So NASA announced about 2 years ago that with all the telescopes that are coming online, including the James Webb Telescope, that NASA thinks by the end of this decade, not that far from now, by the end of this decade, they might be able to generate an exabyte of data every day. Every day. Right? Now, there's something magical about NASA's exabytes already. NASA already admits they generate so many pictures of the sky that there are not enough astronomers on this planet to look at them all. How do you know this? Because NASA set up something called Galaxy Zoo, where you can go as a citizen scientist to take a test, and then you become the one that assigns, is that blob in the sky a star or a galaxy? Now, something very telling about that approach. At a certain point, a field gets so much scientific data, they run out of scientists. And what that means is you have to then take that data to a new community, a new audience. Citizen scientists, crowdsourcer, and you can guess, I'm going to tell you, we're already there in biology and medicine. But indeed, people say that we have so much data now that science itself is going to get obsolete. Now, how do you say science will get obsolete? That's kind of ridiculous. Well, this is Chris Anderson, who's the editor-in-chief of Wired at the time, back in 2008, wrote this very influential paper, influential to me. Now, Wired isn't the most scientific of journals, but I love this one, right? It's not that science is going to get obsolete, the scientific method is getting obsolete. Now, think about the scientific method, right? We're taught this in 3rd or 4th grade usually. The scientific method is you come up with a question, you call it a hypothesis, and then you go gather data to answer the question, right? You know, in 3rd grade, it's like measuring pea shoots growing in soil or whatever, right? Question first, then gather data. But in today's world, we already have the data. 99% of the hard part now is figuring out what's the question I want to ask. What's the killer question that everyone's been dying to know the answer to, maybe for 100 years. And no one's realized you can ask and answer that question right now with the given data. That's the hard part. Look, it's hard to write code and cloud computing and mobile. I get all that. That's not the hard part. The hard part is figuring out what's the tractable question, the killer question, and know that you can answer it with writing code. You can't just do this as a programmer. You got to know what the unmet need is still too. You're going to see this theme a lot. The question is first. We don't write a line of code in my lab unless we know why we're doing this. What's the question we're going to ask? Right? This is the hard part now. And we're certainly drowning in data in life science research, right? We're drowning because of all the gadgets. Now, I could have shown you a sequencer with blinking lights. I could show you a mass spectrometer. I could show you a flow cytometer. I will show you these microarrays because this is how I got my start in Zach's lab back in 1998. These chips were priceless on the left. We had 60 of them, 60 different cancer cell lines. Boy, we were lucky to get that data, right? Those chips were priceless. Now they're so cheap, I carry one in every suit. And this is what they actually look like here, right? So this is, if you're looking, I have it in my hands here. So this is what they physically look like. And it's a good reminder that big data comes from little packages, just like this, right? Data is kind of ethereal, so this is like a physical form of it, right? That's amazing. Everyone started using these chips, right? And, you know, this is 1998, 99, 2000. In fact, we got so tired of measuring them one by one, we started measuring them on the 96 well plate format. That's so cheap, now I carry that in my bag. And then we started using these, and we got tired of measuring them 96 at a time. We switched to the 384 well plate format. And that's so cheap now, I can carry that in my bag. I cannot more clearly illustrate exponential growth in biomedical data and show you these plates. We don't even use this one anymore because we switched to RNA-seq. In fact, this company doesn't exist anymore. Affymetrix got purchased by Thermo Fisher or something, right, last year. This is just what we keep doing in medicine and biology, right? We just keep making more and more measurements. And what do people do? They get a grant, they go make some measurements, they write a paper, and what do they do again? They get another grant to go make more measurements. And what happened? What was magical here? What happened in 2000, 2001, 2002? Everyone started writing papers with these gene chips, right? And again, I'm talking about chips, but it's RNA-seq, it's the RNA side, right? That's how I got my start. There's a list of genes that change our heart failure and cancer and diabetes. Everyone had a list of genes. And it started with the journals, the top-tier journals. They said, you know what, we're getting too many of these papers. Too many papers with gene chips. No more papers with gene chips. Unless you put that data into an international repository where people can double-check all the math. It's probably getting important to do that as we get closer to the clinic. And maybe others can use these samples for something else. The precedent for this goes back to GenBank, right? Everyone's seen GenBank. It's a next door neighbor to PubMed, you know, one of the tabs up there. GenBank, I think, is 48 years old now, right? It's way early, way older than the World Wide Web. They were sending tapes to each other, right? Anyone remember DVDs, CDs? Anyone remember 3-inch discs, 5-inch discs, 8-inch discs? Tapes were before all that, right? Because you read AGTCs in a paper, nobody wants to type in AGTC again, right? They were sending tapes. But the precedent has been set. If enough scientists use a molecular modality, they got to share the data at some point. That's what happened in this field, right? So you got to share the data. Fast forward to August of 2012, when this article came out in Nature, featured my lab and a bunch of others. August of 2012, we hit 1 million samples publicly available, 1 million microsamples, 1 million biopsies, cancer cell lines or other cell lines, mouse models, operating room specimens, 1 million samples publicly available. I started Zach's lab 0 to 1 million, right? Now, if you got really sharp eyes, you see, I mean, this array expresses the Europeans, Geos, the NIH doesn't really matter anymore. They're the same thing. You look really closely, though, it doesn't look like it's slowing down here. It's kind of sad at the end, slowing down. Then you realize they only counted half this year, right? This came out in August of 2012, right? They're still doubling every 4 to 5 years. In fact, if you go right now to Geo, I think it's at 2.1 million samples, right? So there's 1 million. 2 million would be about here. 2018 is here. Straight up, right? Straight up growth. What does this mean? Any researcher today that wants to start a project on colon cancer or diabetes, you don't have to start with animal models. You don't have to start with recruiting patients. You start with downloading the data. Or, like I love to say in my talks all the time, even a high school kid that wants to do a science fair project. Let's say she wants to do a science fair project on breast cancer. She can go to these websites, type in breast cancer, hit search, and find and download 84,000 samples of breast cancer. About as easy as she can find a song on iTunes today. 84,000 samples of breast cancer. What's so magical about 84,000 samples of breast cancer? If you haven't figured it out, that's more samples of breast cancer that high school kid can get to than any researcher in the field of breast cancer. Because every one of them eventually has to submit data into the repository. And whoever figures out the repository now has more data than any researcher in the field. Maybe it's not breast cancer, it's colon cancer, it's prostate cancer. Whatever disease probably has been studied endlessly in the last 18 years with this kind of technology, right? Now, the number I like is 3720. Let's call that 3,700. Let's round it to 3,700. That number basically means on the order of 3,700 different research groups and labs have contributed data on breast cancer to this repository. Let me tell you what a ridiculous number that is. Let's pretend I'm writing a brand new NIH grant on breast cancer, and I'm a newbie. I know nothing about breast cancer. You're all my study section. Let's write the grant. Tell me what you think. I propose to you that I can get 3,700 of the best breast cancer labs in the world to get their best characterized patients, to run the latest measurement technologies. I propose to you I can get all 3,700 of them to share data with me for free. For free. You're already laughing, some of you. If I actually wrote that in an NIH grant, you would laugh me out of the room. Why would 3,700 of the best funded, best breast cancer labs ever share any data with me as a newbie researcher? Yet here it is, sitting there, waiting for you. All this data. When I think of public big data like this, I actually call it retroactive crowdsourcing, right? What does that mean? These 3,700 labs are there to help you on the internet, and they don't even know they're helping you. Actually, these 3,700 labs are there to help you on the internet, and you don't even know they're helping you. And I don't know, maybe 84,000 samples are still not enough for you. In another couple of months, it'll be 100,000 samples, right? In this hour, we'll add more than 200 samples to Geo, just sitting here. Someone in the world is uploading data, right? We don't even have to touch our pipetter right now. All this data is sitting there waiting for you. That's the magic. Now, if you have any doubts, a high school kid can do it. Here's a high school child that did this, right? This is Brittany Wenger from Sarasota, Florida. I've never met her. Talk about her all the time. She went to Duke as an undergrad. She's probably in grad school now. At age 17, she won the top science prize in the Google Science Fair competition because she built an artificial brain to diagnose breast cancer. She downloaded qPCR data, immunohistochemistry data, and figured out how to do a machine learning type of thing and came up with a diagnostic. I guess she was bored. The following year, she was in the paper all over again for doing this for leukemia. Of course, what this means is if a high school kid can do this, Everyone in science better know how to do this. You don't have to be an informatician to do this. Everyone's got to know how to do this. Now let's just highlight a couple important databases for some of the junior folks in the room here. The Cancer Genome Atlas, I'm sure many of you have seen this. It's a very cancer-rich campus here in terms of informatics. We love this data set. 14,000 cases of 39 different kinds of cancers. You can see all the distributions here. And it's not just RNA, 13 kinds of data. It might be oncogene sequencing, so a little bit of proteomics, certainly RNA data and RNA-seq, but also the medical imaging, mammography, some head imaging, clinical data, all that data in this repository. The NIH Links Program, I'll talk about this one a bit. This is an exercise that's funded by NIH. It mostly goes to the Broad Institute that collects, that's something called the connectivity map. Now this is called Links, where they've tested thousands of compounds and thousands of perturbagens, either overexpression or knockdown constructs on a dozen or so cell types. You know, just that grid is 1 million samples right there. And each one of these is a before and after. What does the genome look like? And PubChem. Show of hands, how many of you have heard of PubChem? Yeah, it's about 10 people here or so out of an audience of about 100 or so. That's more than usual. PubChem, I still can't believe they give this out for free. So what's PubChem, right? If, you know, we have cells that glow, right? If you can get your cancer to glow, if it's living or dying, once you get it to glow, that means it's colorimetric. You can put them on spots and get robots to test 1 million drugs or 1,000 drugs. But if NIH funded you to do this, you got to deposit that screening data into PubChem. So think of what is PubChem now. Think of a big grid of 250 million compounds. Those are my columns. More than 1 million assays. Those are my rows. In this grid are 300 trillion little boxes, of which 1 billion of them have been measured. This compound in this assay, this compound in this assay. Now, the 250 million compounds, 71 million meet Lipinski's rule of 5, so it might be orally active. And of the billion measurements, 1 million of them were active. That drug worked in that screen, right? Now, I can easily bet you a beer. This beats any pharmaceutical company's screening data. I can easily bet you a bottle of wine. This beats all of them combined, because this is still doubling every 2 or 3 or 4 years. And very little in pharma discovery is doubling every 3 to 4 to 5 years. And they give out this data without a user ID and password. Probably many future drugs are just sitting there as a column in this grid, waiting for you. If you don't do this, nobody's going to. Because the people who collect this data go get another grant to go collect more data. And all this data is just sitting there, waiting for people like you and me. All right, so you see what high school kids can do with this. What do we want to do? This is get to precision medicine, right? What's precision medicine, right? I go off the 2011 National Academies report. I mean, that's like our Bible at UCSF now, right? Precision medicine is I'm going to customize the medical care I'm going to deliver to you based on measurements I'm going to get from you. Of course, there are going to be biological measurements like DNA and proteins, but I also need to figure out behavioral measurements, right? I might get those from smartphones or questionnaires or your fitness, wearables. I also need to know your preferences. I can tolerate this kind of side effect. I really don't want that kind of side effect, right? I got to get a lot of measurements from you, but I can't just do it with your measurements. I need those kind of measurements on a lot of people, right? As much of a population as I can get to. And I got to know what medical care I've delivered to this population. I figure out what's working or not and then apply it to you, right? So it can't be just personalized. I got to get a whole bunch of people to figure this out. If you look at the original report and the way I think about it, how I structure what I think about in my lab, it comes down to 3 challenges. We need better diagnostics. We need better treatments. We need to figure out our outcomes. Well, it's kind of a no-brainer. I'm going to spend the whole rest of my time just giving you our anecdotes of what we're doing to get to diagnostics, treatments, and outcomes. That's all of them. All right. So let's talk about diagnostics first. So my first big NIH grant, back when I was at Stanford, we were looking at these repositories, right? Boy, at that time, we had a whopping 25,000 publicly available samples. Now we got 2 million. What are we going to do with 25,000? Well, the first grant I got was go get every human disease studied by microbes. Why pick diabetes or cancer? Just go get all the diseases. And we started to collect experiments like this, where in the same experiment, people looked at disease and healthy in the same experiment. Now, disease is actually kind of easy to figure out. Healthy is really hard. We have 200 words for normal in biology. Normal, vehicle, wild type, time zero, margins, control are the first 6. There's like 100 more, right? We had to write a paper explaining the 200 words we use to get all our text mining to work, right? But we human eyeball this stuff. Each box is a gene, and there's more of a gene, less of a gene. People have been calculating signatures like this for decades. Not a big deal. And Marina started this work in my lab, and she's an assistant professor now on her own at UCSF. But you know, for every disease, I don't just have one data set. I might have dozens. For breast cancer, I have thousands of data sets. There might be biopsies. There might be cell lines. There might be animal models. I can look at all of these and try to figure out which ones I want to keep, which ones I want to discard, and figure out what's in common. So that's now, let's go a little bit deeper for a second. As you know, I love these arrays, but they get us to RNA levels are measured here. I want to make diagnostics out of this. Well, what is a diagnostic? Diagnostic helps a medical doctor tell if you have a disease or not, right? We might have imaging diagnostics. I love protein diagnostics because I remember way back when I was in pediatrics clinic, if I had a toddler with a sore throat and a fever screaming their head off on the exam table, somehow I can stick this Q-tip back there, put some drops on it, and it can tell me if the kid has strep throat or not. That's called an ELISA, enzyme-linked immunosorbent assay. That's 1970s proteomics. I love ELISAs because you don't even need a refrigerator for those things to work, right? They work in a desert, right? No cold chain needed, really. But ELISAs, you got to really make them specific against something, right? You can't just make ELISA on everything. You got to know what to build it against. So here's the kind of concept. Remember central dogma, DNA to DNA to RNA to protein. If the RNA is changing in the disease, maybe the protein is too. We're going to detect that using these ELISAs on fluids that we can get. All right, we did a whole bunch of these, and the disease I'll spend a couple minutes talking about is this one, preeclampsia. Show of hands if you've heard of preeclampsia. Yeah, so it's about a third of the audience. It usually is a third of the audience. This is a third of the audience that watches Downton Abbey because one of the characters dies of this disorder. Spoiler alert if you haven't seen the series, right? Sybil, right? Eclampsia. When you're pregnant, you get high blood pressure, you can lead to seizure, the mom dies, the baby dies. Yeah, some of you nodding your head, you've watched this. And this is still how husbands lose wives around the world, right? This is still a major problem. Preeclampsia happens earlier, right? You don't want to get to full eclampsia. And what happens is we have to take these babies out with the mom and mag sulfate. Typically, the mom gets better with the treatment, but then we pediatricians have to keep these tiny babies alive and healthy for months while they're growing until they can go home. 4 drugs in trials in the United States right now for preeclampsia. But the diagnostic we're using is ancient. We look for urine protein, not even a specific one, right? Probably the most non-specific test we have in today's obese America, where a lot of people actually now have protein in the urine. So we wanted to come up with a specific blood test for preeclampsia. Linda Liu was a grad student who got the ball rolling. Bruce Ling did proteomics, and Matt Cooper got involved, as you'll see in a moment. And how do you start? You don't start with pipetters and IRB. You start by typing in preeclampsia in the databases, right? So here we've typed in preterm birth and preeclampsia. 266 experiments that might be relevant. I don't mean 266 chips. 266 collections of chips, right? 299 human samples here, 249 here, 154 here, 143 here. Oh, here's preeclampsia, here's prematurity. You can pick and choose. And now here's the fun part, right? I love that these scientists are sharing their data like they're supposed to. Not everyone does still. I love that they're sharing their data. I love their data sets. I don't trust a single one of them. But I trust what I get in common. What do they all see, right? What a whole bunch of them see, right? Safety in numbers, right? That kind of concept here, wisdom of the crowd. So if I see something in one of the data sets, yeah, okay, but if many of them are seeing it, I'm going to chase it down. And so all of these experiments, think of a big Venn diagram. What are we getting to in the middle that all these people are seeing? If the RNA is upregulated, then I'm going to chase it down as a protein. And so here's an example, one of a dozen that we found called hemopexin, upregulated in preeclampsia compared to normal, healthy pregnant women down across the station. There's Marchadime's funding for this, as well as Spark, which is an internal CTSA seed grant. We wrote a whole bunch of papers. You can chase them down at this link. And what do you do next in Silicon Valley? You start a company. It is not taboo to talk about companies in academia. Shouldn't be, right? Shouldn't be. Here's the arrow. You're going to see a couple of these arrows. We start with the unmet need. We're not going to write a single line of code unless we know what we're trying to answer. Then we get to a diagnostic. Data's available. Marchadime funds the early work. Design the diagnostic with test samples from the freezers and refrigerators. Get it to work with $50,000. Launch the company, $2 million in seed financing. Now, let me be crystal clear for this academic audience. I'm not showing you $2 million to brag about it. It's actually not a lot of money to raise in Silicon Valley, it turns out, if you read the headline. I am showing you the $2 million to maybe convince you this is the new way science has to continue out of our labs. We got the grants, we got the papers. The next scientific experiment that needs to be run is a prospective multi-center trial. Does this marker or these markers actually predict the future for these patients? But that scientific experiment now will be done by a startup company funded by private dollars because it's that much more relevant to patients. Or to put it another way, do you know how hard it is today to get a brand new $2 million NIH grant? The science can continue in the startup companies funded by private dollars. You got to follow all the rules, but that's the concept. So I love this startup because as you can see on the right here, it's already been acquired. Some of you know what that means. So we went from public data, which people think is as valueless as kitten videos, to design a diagnostic, testing diagnostic, starting the company, selling the company, start to finish, 24 months. Inventors happy, investors happy. Of course, we're going to do more of these, and I'm giving away the secrets here. Because every one of you could take a different disease with this approach, and we'd never step on each other's toes. That's how many diagnostics we need in medicine. Every bit of this story are in the links. You just look this up. You can find all the press releases, all the papers are out there. Every step is documented here if you want to go. Talk about another diagnostic, sequencing, right? This is another game changer, right? Obviously, the Genome Project was complete over 13 years, $3 billion, as we all know, in centers like the Whitehead Institute. Now, this is my hand holding one of these new Oxford nanopore devices, the mini ion. I mean, look at this. It's a no-brainer this is going to be used in medicine. I mean, it's only a matter of time before we really use sequencing more and more. And so I'm not a geneticist. I didn't get involved with genetics until very late in the process, around 2009, 2010, because of this individual, Steve Quake. He showed up at Stanford. Now, he's a weird patient. He's actually a Stanford bioengineering faculty member. He starts sequencing companies on the side. But he showed up one day as a patient, the first patient to show up with his whole genome. Because here's Steve here at age 40. His nephew died one day at age 19, sudden cardiac death. One morning, just did not wait. Steve Quake goes to his friend and our friend, Ewan Ashley, cardiologist, and says, well, doctor, am I going to die of sudden death? Here's my genome, doctor. And this was in 2009, okay? This is the early days. This is where they came up with the concept, you know, $1,000 genome, $1 million interpretation. A team of about 40, 50 people got involved in analyzing Steve Quake's. Ewan Ashley looked at the heart. To make a very long story short, he could give you an hour-long talk. There are no known variants in known sudden risk diseases, genes, or set up for arrhythmia in Steve Quake. He has a variant in genes that might set up for myocardial infarction later in life, like many of his family members do, but that's a different kind of sudden death that's later in life. So his heart seemed like it was okay. Russ Altman could give an hour-long talk on the pharmacogenomics here, right? Long story short, 150 drugs you can say something about, as many of you know now. Is it going to work? Could there be side effects? What would the dose be for about 150 drugs? And then my lab was stuck with all the rest of medicine, okay? Heart, check. Pharmacogenes, check. Everything else in medicine came to my lab. Now, to be honest with you, this paper came out in 2010, and we got the genome in 2009. We were kind of thinking this was going to happen someday. In 2007, I realized that a patient was going to show up someday, and we were totally unprepared for this at Stanford. Because there was no master list of just every SNP, every variant in every disease. Right? There's no master list. And if you say the GWAS catalog, I'll tell you the problems with the GWAS catalog. Okay, I love the GWAS catalog, but for a third of the alleles, they don't even tell you which was the risk allele, which was the protective allele. That's useless. We need to know every allele, everything, right? So we just started with a San Jose high school kid. I had him for the summer, gave him 50 papers to curate. I said, just read these top 50 papers and see what you can extract. What's the meat you can get out of these papers? So what does it mean to read a genetics paper? Here's an example of a genetics paper. This is a paper published in 2008. It's one of the ones we've read in a journal called Inflammatory Bowel Disease. With a journal name like that, you can guess what kind of diseases they talk about in that journal, right? Crohn's disease, ulcerative colitis. They looked at 9 loci. So this is an example of a post-GWAS paper, 700 finished patients. So a paper like this, we record 100 different items. GWAS or non-GWAS, what kind of structure was it? disease and phenotypes. So disease would be type 2 diabetes, phenotypes like fasting blood sugar. The population, gender, boy, you know, and Gonzalo's still in the room here somewhere, so he knows this. You know, there's no master list of just every ethnicity in the world. We have a master list of 863 ethnicities that we found in papers, right? We just made it up, right? There's no list. All the alleles, genotypes, p-values, odd ratios, all the confidence intervals, all the technologies, and we mapped all this to UMLS concepts. So just to give you an example, here's a table from this paper, and this says RS1004819, that's a DB SNP allele. Humans have a C and a T, and we write down every one of these numbers. And then we back calculate how many people were in each little bucket, and we keep track of all those numbers. Does that make sense? Look, I love NLP. Yeah, I'm a computer science guy. You know NLP can never do this. And if you want proof of that, take this PDF, open it in Acrobat, and just select this table and hit copy. Because Acrobat doesn't even know where to put the tabs at this point, right? Forget about trying to figure out which column is which. You can't even get the tabs in the right place when you copy from the PDF, right? So you got to read every supplemental table, everything, right? You can't get this from the abstract. All right, here's another paper. This one came out a year later in a journal called Rheumatology. You can kind of guess what diseases they have there. Ankylosing spondylitis. This one came from the Wellcome Trust. This was a post-GWAS paper. 8 SNPs, IL-23 receptor in 2,000 UK case control patients. This table is sideways. You have to rotate it. We write down all the numbers. Here's an example. The first SNP here is RS1004819. Humans have an A, and we get this all down. Now, if you were paying attention, you would realize I just showed you the same SNP in 2 papers. And this paper says humans have a C and a T, and this paper says there's an A. What are the alleles for this? And then you got to realize you got to chase every one of them down in DB SNP. The alleles are C and T. Wellcome Trust got this one wrong. And then you realize 11% of all papers published in human genetics get this wrong. It's not a strand problem. They're reporting on the probe they used, right? That allele's in neither strand, right? Now you got to fix all of these. That's what we did. We just literally then calculated if you can do 50 with a high school kid in a summer, what does it take to read the entire genetics literature? That's what we did. Here's 1 paper. Here's 6 papers. Here's 100 papers. Here's 1,000 papers. So I literally have a Python script that you can do this with PDFs. Make a long story short, Rong Chen ran this with an outsourcing company because we can just hire geneticists, bioinformaticians in India to just read these papers. We have a team of 100 people reading, right? And then we made it to, we've blown, this is an old slide, I don't even know the latest ones. We made it to 20,000 papers, 1.6 million records of 500,000 SNPs, 7,400 different diseases or phenotypes. We call this VariMed. Now, actually, we're going to give out a good chunk of this. Now, this got involved with the company. I'll explain a little bit that in a second, but we're going to actually open source about 1/3 of this database now, the part that we did while we were. Now, what do we do with this data? We start to make diagrams like this, calculating the risk. For Steve Quake, you know, these are all the diseases we think we can calculate he's at risk for. And we're using, I'm not going to go into details here, instead of odds ratios, which geneticists like to use, we actually back calculated what are called likelihood ratios, because that's what medical professionals are trained on. So because we had all the numbers, we could calculate LR instead of OR. But more importantly, on the edge here are all the environmental factors. We mined the hell out of PubMed and these mesh subcategories. We're looking for environmental factors that actually trigger the etiology of these diseases. And we think this could be a prescription pad of the future. This is what you should go after for changing your behavior. So, Steve Quake, you should watch your diet. You should exercise. You should not smoke. You should think about limiting your alcohol. Probably don't need a genome for those 4. I admit. But in tiny font up there are pesticides and Parkinson's disease. And 10 papers have linked pesticides to the etiology of Parkinson's. I don't write the papers. That's what PubMed has. So, Steve Quake, if you like to garden, just watch out for the pesticides, right? And maybe this is now how doctors and patients can talk about their genomes. This paper was a prototype here. Not with AGCs and Cs and RSIDs. You're doing great with stress. Come back in 6 months, work on your diet, and all these numbers and odds change, right? Because it's changing as we get older. You're not going to get childhood stuff. You might get more of the older stuff, right? This is how I think we can communicate with that. Certainly, genomes are used in many different ways now. We got involved with a couple of these kids. The first was obviously at Milwaukee. Nick Volker. If you don't know this story, you got to go read this story. Because now kids with an N of 1 can come to our children's hospitals, get sequenced. We actually can have an explanation for a lot of these kids. Syndromes. This was a kid that was going to die. He's literally the poster child for DNA sequencing. Pulitzer Prize winning story. I'm not going to spend more time there. This is really their story, great story. And just look up Nick Volker when you have time. It's a great story. It's really a reason why. And of course, we're doing cancer and neurological diseases. And what do you do next in Silicon Valley with all of this and other faculty to get started? You start a company. And this became Personalis. With our database, Russ's database with pharmacogenes, Ewan with rare variants, Mike Snyder with all the intergenic stuff. We have this need to predict the diseases. Publications were there. We got Stanford money to pay for all these curators. And to make a long story short, we've raised $76 million now from investors. And to put it really simply, do you know how hard it is to get a $76 million NIH grant? The science continues in the startup companies, right? But it's more relevant to patients. That's why it's going to be better to do it there instead of us trying to get that kind of thing. We got the papers, we got the grants. Moving on now to the next step. Let's talk about the third one, drugs. Drugs are a much harder story. How are we going to get to drugs? If I ask you how much does it cost to develop a drug, most people answer it's $1 billion in 10 years to make a new drug, right? You've heard that? $1 billion in 10 years to make a new drug? Boy, that's an underestimate. It's an underestimate. How much does it cost to really make a drug? Well, Matthew Herper does this every couple of years in Forbes. It's really simple math, even for this late in the afternoon. How much did you spend? Divide by how many drugs did you get? Yeah, real simple math. Do that math for the top 12 pharma companies. They spend between $4 billion and $12 billion per drug. Kind of not sustainable today. Now, that's a really negative way to pick on the pharma industry. Let's paint this in a more positive way. Even if every pharma and biotech company on the planet was 100% successful in developing the drugs they want to develop, there are not enough of those companies on the planet to generate all the drugs we're going to need for precision medicine. There're just not enough companies. It's a NASA astronomer problem all over again. There's just not enough astronomers. There are not enough companies. We have to come up with a more efficient way to do this. What I realize, it's going to come down to us in academia to solve this. I love the pharma and biotech industry. I consult for them all. You saw my second slide. I have no faith that they're going to be able to solve this problem. The culture is just not exactly right there in those companies. It's going to come down to us to figure this out. We got to do this. Now, I'm not saying I have the world's solution to this. I'm just going to show you how we do this, right? And how are we going to do this? Well, we're going to use data, right? So I told you we could get to diseases and healthy samples on the left. On the right, we start to realize people dumping drug data onto the internet. I showed you links and connectivity map. I showed you PubChem. Amazingly, a lot of times they're using the same exact assays as we're using on the left. So if we got disease and no disease on the left and cells with and without drugs on the right, we're going to put these two together. The reporters call this Match.com for drugs. You know Match.com, right? You get a date or a spouse, maybe if you're lucky, I guess, or a mate, right? What's the famous saying if you're trying to find a spouse? Opposites attract, right? You remember that? You've heard of that? Opposites attract? I'm going to show you all of the bioinformatics involved with my arms. If I got a disease on the left where this gene goes up and this gene goes down, and I can find a drug now that knows how to make this one go down and this one go up, maybe there's a match there, right? That's two genes. Imagine 20,000 genes. Imagine Kolmogorov-Smirnov tests and Pearson correlation coefficients. I don't need to show you any of that. I'm just naively reversing the pattern I see in the disease. Really simple concept. Right? We turn the crank. We got lots of these to work, right? Here's a new drug, and there's a new drug. Where we got a lot of traction aren't the new drugs. It's the new uses of the old drugs, drug repositioning. Now, we didn't invent the concept of drug repositioning. Everyone knows the classic examples. The old cardiac drug that had an interesting side effect of hair growth. That's minoxidil today. Of course, everyone knows the other cardiac drug that had its own interesting side effects, right? And the patients don't want to give it back. That's Viagra, of course. That started as an angina drug. Now, Viagra has done a double reposition. It's also known as Revatio. It's used for pediatric pulmonary hypertension, right? It opens those blood vessels from the heart to lungs in babies where it's too tight there. Everyone's teasing about babies on Viagra, you know. But you know what? Instead of finding these by accident, how about we find them on purpose using public data, right? That's the all right. We turn our crank, we got lots of these ideas. Here's a drug, and there's a drug. How are you going to know this works? You're going to have to run a clinical trial. Oh, hell, nobody's going to put this into patients until you test this in an animal model first, right? You got to run a preclinical assay. Boy, all my mouse friends, they scattered. We had a great number of friends that helped us with a lot of this work, you know, testing these drugs. But now, what changed my life? Now, when I really need to know if the drug's going to work in disease, I just go to assaydepot.com. Kind of sounds like Home Depot. And you'd be right. Let's go browsing in the store. These are my aisles. Biology, chemistry. DMPK stands for drug metabolism pharmacokinetics. If you don't know what that stands for, don't click there. Pharmacology, toxicology. All right, let's click on pharmacology. We know what that means. What type of mouse do you want to run today? Bone, cardiovascular, dermatology, diabetes, venereal, infectious disease, inflammation, neurology, cancer, eye, ear, pain, respiratory. I don't know. Once in a while, I'm a diabetes guy. Technically, I'm an endocrinologist. Let's click on diabetes. What do you see? Oh, here, I can now order a 16 mouse study. You know, maybe I have 2 groups of 8 with and without drug, or 4 groups of 4, you know, dose ranging study. OB diabetes model. What is OB? If you read here, it says this mouse has been eating a lot and had diabetes since 1951. This is more or less a standard model of diabetes. It is neither the best one nor the worst one. It's one that many people use. And in 28 days, I can get back all the insulin tolerance tests, fasting blood sugar, glucose tolerance tests. I send a drug blinded, and I'm going to get back all the answers. What am I covering up here? The price, $9,000 for this service, 9-week turnaround time. I kid you not. Add to shopping. I can buy an entire mouse model now off the internet with a credit card. What the hell is this, you're asking? These are called contract research organizations. CROs have been around forever. Pharma uses them. But because of Science Exchange, Assay Depot, now they're called Scientist.com, because of transparency in clearinghouses, the pricing has come down that we can even afford this in academia, right? Now, let me be clear for a second, right? Some of you might be mouse researchers who work in mouse labs. Those PIs would look at $9,000 for 16 mice. That's ridiculously expensive because they have slaves. I mean, graduate students that can do this work for so cheap. But for dry bench researchers like me, what does MasterCard say? This is price. There's no way they'll ever let me into that house. And I can pay a company to do this. Now, some of you are really concerned, right? What about the quality of this experiment? Well, actually, when you hit add to shopping cart, it shows you the 133 labs or companies that are willing to do this. It's not just one, right? Including University of Maryland, Wisconsin. Outsourcing doesn't mean offshoring. You can pick and choose wherever you want to do it. Let's say you need them to have certifications, right? I need them to be ISO 9001. There's 7 of them. FDA 5, triple ELAC 28. You click on certifications, you're only going to see the companies that are certified. It might cost you more. I can get whatever certifications I want. Boy, I wish it were that easy to find the right collaborator on my campus where I knew someone with certifications was doing the experiment, not some random undergrad, right? I mean, I wish you could do that even inside our. But some of you are really still hyper concerned here. Because you know, when this works, I'm going to humans next. How do you trust the results of a company like this? And I would just simply say, if I really, really, really need to know if this drug is going to work in this mouse model, I just order 2 of them from 2 companies. Hell, at $9,000 each, I can order 10 of them from 10 companies. Now, why am I saying it like this? Think about a mouse researcher who gets a mouse experiment to work. Do they go across the hall to get a completely independent team of researchers to repeat the experiment? You know nobody does that. But I can hire as many independent teams as I want if I have enough money. And now what I can do is take the quality problem in your head and make it a function of dollars. I have more dollars, I can hire more teams. Don't have a lot of dollars, maybe I'll just hire 1 or 2. Now, we typically will just hire 1 from the West Coast, 1 from the East Coast. I have nothing against flyover states at all. We'll be better at ordering from the Midwest here, right? But typically it's the Bay Area and the Boston areas where we order from, right? That's just how we can do this. Now, we've done this like crazy in our lab. Of course, we still use core labs and collaborators, but what's the beauty here, right? The beauty is I can work on 1 disease 1 week and switch to another disease the next week. No mouse research can do. They got to love their mice. They got to treat them and take care of them. I can switch to another model just in a 1 week's notice, right? That's the beauty of doing this. We did a bunch of these. The one that got us on the front page of Wall Street Journal was this one. This was in 2011, Science Translational Medicine, where we predicted an epilepsy drug, topiramate, would treat inflammatory bowel disease. This is a normal. So these are my famous rat colonoscopy videos. Not enough people are laughing. So let's illustrate the geometry involved, right? This is a colonoscope. This is a rat. Why would you ever want to know how to do this in your lab, right? There's a company that knows how to do this? Outsource, right? This is a normal rat colon, rat with this a chemical called TNBS that makes the rat get IBD. All better with the pyramid. 20, I think 40 megs of video at the Science Translational Medicine website, right? We've done a bunch of these here, right? Here's one that came out last summer. I like this one. This is called liver cancer, hepatocellular carcinoma. 350,000 patients a year get liver cancer in the world, but very few in the United States. So there are not a lot of companies making drugs on hepatocellular carcinoma because this is a hepatitis problem and alcoholism. It's just not that common in the US, not a lot of companies working on it. It's mostly India and China. Here we predicted this drug, nicolosamide. We give this for worms in your GI tract. Talk about wiping out that microbiome. Like nicolosamide, you got worms, this gets rid of them. Control, no drug, nicolosamide, yeah, kind of better. Nicolosamide ethylolamide is a particular salt form that is orally absorbed. So we can give this in the chow for these mice. This is a xenograft, human cancer in the liver model and a mouse model. Glowing cancers, no drug with the nicolosamide. You can see a couple of mice doing better here. Now we do have a drug already for liver cancer. It's called sorafenib. It's not that old drug, but it's also not that good. You can test that and this drug together. Here's no drug, nicolosamide, sorafenib, and together we see synergy, which means nicolosamide is probably working in a different way than sorafenib. So when we launch the clinical trials here, this is going to be how we do it. And this is just sitting there, nicolosamide and its prediction. This just came out. All right, let's show some new data. This isn't published yet. This is a really cool one. Psoriasis. Somebody over there knows about psoriasis in this room because there was a poster on psoriasis, right? Psoriasis is like thickening of the skin, you know, like it's an autoimmune condition, thickening, plaquey kind of skin. And so this mouse has been genetically engineered to get psoriasis, particular overexpression construct of a transcription factor. Mazen did the work in my lab. Peter and Martin did the work there with the mouse. Look, I'm neither a mouse person nor a psoriasis person. I don't know, the hair looks kind of weird here. I can't really see the fingers here. That's before. This is after. It looks a little bit cuter. I see the fingers here, right? What's this drug? This is a 1960s diuretic. We used to use this drug in the 1960s to help you pee. We have much better ways to make you pee now. We don't use this drug anymore, okay? Now, this drug is kind of interesting. Whether you give it systemically or topically, the psoriasis is all better. Now, you could argue back, why are you working at psoriasis? Biotech companies, they'd love to sell you an anti-IL-12/23 monoclonal antibody at $50,000 a year. It's an immunosuppressant. You wipe out good components of the immune system. Yeah, the psoriasis gets better, right? That's what we do. Use monoclonal antibodies. Let me show you the cellular data. On the top left is normal wild-type skin. This is the pink are the keratinocytes. Okay, you see them at the top there. This is the model. Keratinocytes go crazy deep. Instead of staying at the top, they go deep. It's like growing on a hair follicle, right? They go deep. This is half the dose. It's double dose. You see, the keratinocytes are all back to where they're supposed to be. Kind of neat. The most important part of the slide are the blue. Those are all the immune cells. This is the first small molecule that fixes the keratinocytes in psoriasis and is not an immunosuppressant. Nobody has a molecule like this. And it is sitting there in public data, a 1960s diuretic. Can I more convince you the value of open big data? It's just sitting there. Who knows how many other drugs are just sitting there waiting for you? But for some reason, we think when data's on the internet, we don't want to use it because we've been trained in this ridiculous way. Because if something is free and on the internet, it must be valueless, right? Must be. But this is the raw data of the best scientists in the world sitting there waiting for you. Now, what do you do next in Silicon Valley? You start a company. And this became Numedi, raised more than $10 million. Like a very long story short, these tiny little teams, 4 full-time people in this biotech company. I call these garage biotechs now because we revere garages in Silicon Valley. We worship garages, like Apple and HP started a garage. They're national monuments now, right? We love garages or dorm rooms. We start companies in dorm rooms too. What's the most minimal biotech company you could start in a garage? Enabled with 2 million samples and every mouse model you want on the internet with a credit card, right? That's the concept here. These little teams are making drugs, literally in psoriasis for Allergan, Astellas, and now they're working on idiopathic pulmonary fibrosis, a nasty condition. Nobody really knows why people get IPF. Not the 2 drugs out there, neither is really that good. All right, if you ask me, What's the next big open data that's coming? Where's the hockey puck going that I'm skating to? Clinical trial. What's a clinical trial? The most expensive experiment in the world. And boy, half of them fail. And when they fail, we don't even write papers about it, never mind release the data. Oh man, is that going to change? EMA, which is the European FDA, is now requiring raw clinical trials data released to the public. I don't mean that summary table in clinicaltrials.gov. I mean every patient, every dose, every arm, every encounter, de-identified, of course. FDA might get there if the national academies figure out the politics again and again, because pharma don't necessarily want this to happen. What does it mean to have clinical trials? Yeah, I think we're going to do amazing things. Pharma company, why do you think this failed? This drug seems to work in a quarter of the patients. Here's a marker for that. Let me test that marker in another study, right? Drug repositioning, patient subsetting, drug rescue. I think there's a lot we're going to do with this. I run this repository for NIAID, allergy infectious disease. We call this import. You can go sign up now and download hundreds of raw clinical trials. We're trying to learn what to do with this data. I'm just telling you where the puck is going that I'm skating, right? We do this with Northrop Grumman, amazing bunch of bioinformatics professionals out in Rockville. And now we're getting data from Mark Dimes and Bill and Melinda as well. Sanchita and Liz do all the work on this one. Oh yeah. So now we have so many clinical trials now, right? There's so many clinical trials. You could actually think about not just the intervention arms. Think about all the control groups, right? Untreated control groups. Here's a disease like normal, like with and without disease. So here's patients with a disease, without a disease. Think about all the control groups of just healthy, normal individuals. When we just counted every patient in every control group in every trial that we had, we realized we had more than 10,000 patients. So we've just released and harmonized all the data and called it the 10,000 Immunome Project. Okay, we didn't go and have to measure any immunology in any of these 10,000. It was already measured. We showed how to harmonize all that data. It's already on bioRxiv. You can find the link here. Go to 10kimmunomes.org. You can go download. There's our shiny thing working there. Just go download like crazy. And eventually, the journal will accept it. But who really cares today with bioRxiv, right? You know where I'm going with that, right? So that's just one thing of many that you can do with this. All right. Real privilege now and pleasure to be at this new Institute for Computational Science to get this thing launched. We're slumming it out in this brand new building until our brand new building is built, opening in fall of 2019. If you're coming to Mission Bay in San Francisco, it's a crazy amount of development right now. Just a 2-block radius. This is the Golden State Warriors' new arena. This is our building. They're up to the 2nd floor now. That's where we're going to be. This will be an 11-story building by the end of the next year. And behind here is Illumina and the Chan Zuckerberg Initiative. That's where Chan Zuckerberg Science is. And over on this side, it's the new Precision Medicine Cancer Building and our new Children's, Women, and Cancer Center. Amazing time. I get to run this new institute. And we got launched with this $10 million gift from Priscilla Chan and Mark Zuckerberg. I'm going to show you what we've been spending the money on. So this is how we got our launch. 49 faculty. The vast majority were already scattered across UCSF in all 4 schools, 15 different departments. We're now the academic home for them. You can see all their kind of credentials here. We're academic affinity home for faculty. They're also in other departments. Obviously, research and development keeps spinning out technologies. Educational plans. So this is one point that I want to make here. Of course, we have graduate programs. Many of you are in graduate programs here. We have postdocs that we can train. I took a different route with the educational plans here. I got our institute to partner with a group called Software Carpentry. It's a national nonprofit to teach people how to write code. Basically, we've paid now for enough seats in Software Carpentry to make sure every single UCSF student now learns how to write code. I don't care if you're an immunology grad student or a pharmacy student. If you want to learn how to write code now, we've paid for the seat. Nearly 1,000 students have taken advantage of this in the last 3 years. For heaven's sake, I don't know if you know, in California, in 3rd grade, you know, you used to have to learn how to write cursive. We're getting rid of cursive and replacing it with Python programming now in California. It's about time all of our students learn how to write code, right? And I don't mean the graduate programs. We're all great at the graduate stuff. I mean, this is the tide that's got to raise everyone. So that's what we're doing with educational. Of course, recruit faculty. We're all competing for the same rare unicorn, right? Who knows both computation and biology. We're trying our best to recruit them too. And use our new data assets. And that's what I'll spend the last few minutes on, the data assets. Because what they did, when I got to UCSF, why did I leave Stanford? I love Stanford, never had a bad day. I still lived there near Menlo Park. But they also put me in charge of building a central data warehouse for all patient data in entire University of California system. So just to remind you, University of California has 10 campuses. 6 are medical, 4 are not medical. Like Berkeley is not medical, Santa Cruz is not medical. The other 6 are medical. Of the 6 medical ones, Riverside is brand new. They have a medical school, no real medical system. Let's leave them out for a second. There's 5 left. All 5 have NCI designated cancer centers. All 5 have CTSA programs. So these are probably some of the best, right? And now we get to build a central data warehouse for all that patient data. At last count, that's more than 15 million patients getting care in the University of California. Or if you do the math, about 5% of the US population gets care in the University of California. Now we're building on something called UC Rex. Lucille Ono Machado and a whole bunch of people 7 years ago got I2B2 to work, and we've been having harmonization efforts for more than 7 years. And more importantly, teams that trust each other, right? That know that we're not trying to screw each other on these different campuses. We all get it. We all work together. But distributed and federated databases, as cool as they are, they're only as powerful as the weakest link. When one of those sites is offline, you get nothing done that way. Now we're talking about a central data warehouse. I wouldn't be an IT-ish kind of guy if I didn't show you a boxology slide. Here, 6 campuses, all going into a central clinical data warehouse. What are we going to do with this data? Oh yeah, why are we doing this? Look, I get the national narrative. Everyone says harmonization is hard. Everyone says sharing data is hard. I get that. And academics keep saying we should share data. We even got Joe Biden saying we should share data. You know, it's so much easier for institutions to share data when there's a business reason to share. So I experiment a lot with business models. What we announced last summer is the entirety of UC, what we call University of California Health, UC Health, to partner with United Health Group to make a single accountable care organization for the state of California. Aspirationally, in 5 to 10 years. This isn't going to happen next year. 10-year strategic relationship, but you know, we got to learn from them how to bill, how to take on risk. We're not saying we know how to do this. We got to learn from United to do this, and they get to build up some market share. So it was a win-win opportunity with United Health. And amazingly, all of a sudden, if you're going to build one ACO, we got to know how you practice medicine. You got to know how we practice medicine. How do you take care of a transplant? How do you take care of this? All of a sudden, you got to see everyone's data. You need a data warehouse, right? And all of a sudden, it's much more powerful than actually the academics saying anything. Now, UC Health has amazing superpowers. They're so secret, even we don't know about them sometimes. Most people don't realize, but of our 6 medical systems, if any one of them approves an IRB, the others automatically approve it. It's called IRB reliance. We already have that, right? Scaling times 6 because we have friends on the other campus. In fact, if we sign a contract with Google or Genentech, all the campuses automatically agree to the terms if the Office of the President signs off. It's an unbelievable ability to scale, right? That's what's neat about it. Now, of course, we use this data for a lot of things. We're part of the All of Us cohort. We end up Obama's million or more volunteers. So I think we got this really because we showed we can get our act together in a central data way, and we're part of moonshots and all sorts of things. What are we going to do with this data? This is a complicated slide. Don't try to write it down if anyone's taking notes. I've tweeted this many times. There are 10 bullets here for the 10 campuses of UC. The non-medical ones should get the data too. Clinical researchers someday could do a GWAS if we get the genetic side of it. Community activists, the public might look at disease clusters. App designers could think about using real data to figure out how to counsel patients. Cancer genomics researchers should be able to get all cancer genomes in the UC system. AI researchers should be able to build real data-driven predictors and all the rest here. This is proof that it actually works. This slide is only 6 months old. We've shown it publicly now. This is geocoding of every single active patient in the University of California Health System. 2.1 million of them have shown up in the last 3 years, at least twice. That's one definition of active. And you can see UCSF, UC Davis, UCLA, UC Irvine is this kind of teal color, and UC San Diego is this orange, and then Riverside is here. And by the way, this is Las Vegas, because we have patients there too. In fact, the Hawaiian Islands are covered with our colors. In fact, every city has our patients. When you're really sick, you come to us. We see you. We know where you live, right? It's all demographics, race, ethnicity. This is actually trivial. You can do this with Tableau. This is with Tableau, okay? So you don't even need to write a line of code in R for this. We announced last fall that all the cancer centers, NCI designated comprehensive cancer centers, are going to act as one now, so the Cancer Consortium. And why we're going to do that is because now the entire system can go for clinical trials. And as a model, what we showed is we partnered with AstraZeneca to get a head and neck cancer trial. Head and neck cancer is nasty to have. But the entire UC negotiated for that trial, not PI by PI here, right? Whole system now goes for the trial here. Here are all the cancer patients in the University of California system, seen in 2016. 141,000 cancer patients a year. That blows away when you look collectively, MD Anderson or any of the rest, right? We just have the volume. I mean, all the kinds of cancer and all of that. All right, let's end with what I'm doing in my lab. I'm starting to think about what we're going to do and how we study diseases and what we're doing to treat disease. I'm going to focus on type 2 diabetes for a moment here. This is the graphic as Pastelli. This is a very California graphic. Think about it, right? It's kind of Art Deco-ish. This is the graphic that we use from the American Diabetes Association to treat type 2 diabetes or adult onset diabetes. Now, just mind you for a second, this is a disease where we have a graphic. The vast majority of diseases in medicine, we have no such graphic to know how to treat them. All right, patient comes in with type 2 diabetes. You're supposed to tell them to eat healthy, lose weight, exercise. If and when that fails, you go to metformin. If and when that fails, you add one of these 6 drugs. They're all pastel-y colors up there. When those fail, add the other 5 drugs. When all that fails, just go to metformin and insulin at the bottom. Right? You see that top to the bottom, right? You get the idea, right? All right, so how do I choose one of these 6? Well, the choice depends on a variety of patient and disease-specific factors. In other words, there's really no clue there. Okay? So, simple, stupid question. What do we do at UCSF? Right? When a patient shows up, which one of these do we go into? Why is it important? Because some of these boxes cost 100 times more than some of the other boxes. But they're all shown in the same kind of size and shape, right? So it's an important decision, right? Especially when most pharma trials now are shooting for non-inferiority. No one's so gutsy to say they're better. They just don't want to be worse, right? It's not really even that much better, but they're 100 times more price. What are we actually doing? So I'm going to show you these rings. There are 12,000 patients in this ring. Now, we used to call these diabetes donuts, and they realized that would be inappropriate. So now we call these life-saving. So Tom Peterson is doing this work, right? So think of this as a pie chart. A pie is another inappropriate thing for diabetes, right? But here's a third of patients are on metformin. This is the earliest drug that we can find, we put them on, metformin. A third start on insulin, okay. A couple start on sulfonylureas or glycides. Some start on combinations and more combinations. Some start on very expensive drugs. And the black means there's so many slices there, I can't even show you, right? Each direction out of the center is a different way to treat these patients, 12,000. Now, let me tell you how I'm thinking about diseases. In medicine, it's actually curious. In medicine, we have, the more I've looked at how we and others treat patients with diseases, I think of medicine now as a game, not to belittle having a disease. But think about games like checkers and chess and Go. Now, I got to learn how to play Go, since computers will know how to play Go. I don't know. Chess and checkers, right? You make a move, the opponent makes a move. You make the counter move, they make the counter move, right? I think medicine is like this too. We make a move, and the patient disease makes a move, and we make a counter move, right? So I've made a move now. 90 days later, the patient goes home. 90 days later, they come back. Here's the next move. All of these patients did well. They never had to change the dose. Again, yellow to yellow means we had to change the dose. Yellow to some other color means we added a drug, changed the drug, subtracted the drug, and all the rest. Patient goes home. 90 days later, they come back. Here's the next move. We had to change this or change the dose again. Patient goes home. Patient comes back. Here's the 4th move. I'm showing you 4 moves out in this game, and then you realize we have 1,600 different ways to take care of type 2 diabetes at UCSF. Probably ridiculous, right? Unnecessary practice variation, probably. Now, a couple cool things I can do with this. I have the blood test, the hemoglobin A1C, up and down this graphic, right? What if I did comparative effectiveness of strategies, right? Which are the right ones? Maybe I could get it down to 1,000 or 100 or 10, right? I think there's probably too many here, especially when some of these folks are costing so much more than other folks. And what's really cool is now, University of California, when I want it done 2 days later, we have this for the entire University of California. 71,000 patients with diabetes now. 6,500 ways to treat diabetes. It's worse. It's worse, right? And they look the same. They're really different here, right? We got to get our handle on this. We're going to make an ACO out of this, right? That's what I mean about studying the care trajectories. Now, Tom and Lisa and Ion get to do this, and it's a really amazing work to work with them. I'll skip that one. All right, I'm going to end with this. That's why watch is buzzing. We're almost done. In the end, if you think really far about this, what I want to do is think about maps of death and disease. I want to know how patients today are going to get diseases and die, especially in California. Now think of Google Maps, right? Google Maps takes you to pleasant destinations. I'm trying to figure out if you're in my state how you're going to die. What's the route you're going to take? Perhaps the exact opposite of Google Maps. Here's an example of a map we've learned from real California data. How to read this. Patients show up with alcoholism. A year after that, they show up with liver disease or cirrhosis. A year after that, they show up with liver abscess. You can go straight down. Each arrow is 1 year. The thickness goes along with how many patients go that way. And the squares mean you can die of those conditions. So, you know, it's really hard to show up at a hospital in California and die of alcoholism. But you can die of these conditions, right? So Hannah and Jay have been doing this. Yes, example of a map. Here the map is a little bit more complicated. Patients show up on the left with heart attack. You know what a heart attack is. You can die right away, or you can end up with heart failure. That means the heart is no longer strong enough to pump the blood to the body, right? And you can die of that, or you got lung disease because the fluid starts to back up into the lungs. And then 3 years after the heart attack, we have a lot of patients dying of sepsis on the right. Sepsis is a body-wide infection. Now, let me be clear here. I'm a pediatrician. I didn't really take care of a lot of patients with a heart attack. But I always thought if you had a heart attack, it's the heart that kills you in the end, right? But actually, 3 years later, many are dying from the sepsis, the infection at the end. And you know, if you don't take the northern route, you can take the southern route, kill your kidneys, and end up with sepsis a year earlier, right? And it's kind of weird. We don't really actually even screen or think about sepsis in this way, but it's sitting there waiting if you're not really careful about it. And these are what I mean by maps of death and disease. Now, look, I love Google Maps. I use Google Maps all the time. But there's still one thing Google Maps cannot do. Google Maps cannot show you all the little cars on the map. I know it shows you the traffic, but there's no way for Google Maps to show every little itty-bitty car on the map, right? How could Google Maps ever do that? But when we open our new building in the fall of 2019, we're going to have one room with a wall of monitors, and we're going to have our maps. We don't want to just make our maps. We want to show every single patient on the map. It's a real prototype with real California data. As patients are getting older, they're moving from disease to disease to disease. The colors are getting brighter. They're starting to die now. They're in their 60s and 70s. A whole bunch of them are about to get sepsis and die. There they go. And now we can start to predict what's going to happen in the next 90 days, what's going to happen in the next year, and what are we going to do about it? And that, to me, is going to be the new definition of an accountable care organization. One that knows how to account for the care of 15 million patients. Well, they're dying of earlier diseases, right? Childhood disease. But this is the new definition of accountable care organization. One that knows how to account for the care of 15 million patients. That's what we're building at the University of California. That is literally what we're building. And not just treating patients in counter to counter, but with all the devices and gadgets. Lessons I learned along the way for all the trainees here. I added this at the last one. Bunch of you trainees. What have I learned in the 18 years since I started in Zach's lab? We're only as junior as we want to be. Junior faculty, right? We use this phrase all the time, junior faculty. Junior faculty should be used as a state of funding, not a state of mind. Junior faculty should be trying to kick their mentors' asses, right? Zach loves it when I kick his lab and he kicks mine back, right? We still buy each other drinks. You shouldn't be trying to be under your mentor's shadow. You got to be brighter, always. That's what mentors want us. It should just be a funding thing. You shouldn't be thinking less, right? Set the level of your peers as high as you can. Never think, oh boy, the other students in my program aren't publishing much. Look, I've published enough. Your peers are all over the world in countries you've never been to now, right? That's your new level. If we do not communicate more and share more, we're going to be invisible. You need to be on Twitter. Why are we so critical of fake news? With 2 million publications a year in our field, why are we putting out true news out there? And still, we can criticize fake news and then don't want to be on Twitter. I don't get it. Innovate beyond your university. Get into startups. I think every grad student should just file some intellectual property. That office comes and teaches you what it's all about. You'll never regret learning about it. Shoot for changing the world. Look, I'm an H-index junkie. I'm vain. I'll admit it. In fact, there are fun games, but they're just games. Solve real-world problems, address real-world needs, not just the easy ones, the ones that get you a quick and dirty paper. Our entire field is too small. Not only should every single patient be in a trial, we should be learning from every single patient. We have to cheer our colleagues more to get more total funding into science, not keep putting each other down. You determine your future, not NIH. Why do I show you that? Because I will show you my first career development award, my first K award. When I wrote it with Zach, these are the study section reviews. You can see my first score was a 414. This is when they went from 100 to 500. 500 was the worst possible score. They didn't triage back then. This is as bad as you can get. That's how I started. Why do I say this? It's because sometimes we have this weird dynamic where people say, I'm only going to stay in academia if I get funded. You should be thinking the different way. If I stay in academia, I will eventually get funded, okay? It will take you years today, but it starts with a determination. It doesn't start by some study section telling you what your career is. What is big data in biomedicine in the end? Is it algorithms, programmers, Hadoop, databases, cloud computing, high-performance computing, mobile? Yeah, it's kind of about all these. I showed you none of this. So predicting diseases before they strike, explaining rare diseases that defy experts, finding drugs for disease that lack attention. There are not enough pharma or biotech companies on the planet, making sure we do the right, safe, cost-effective thing for patients. And all this is an amazing platform for biomedical innovation and entrepreneurship. I sum all this up with one word: hope. Patients, families, providers need more drugs and diagnostics. If we don't do this, I don't think anybody's going to do this. We're sitting on this data. It's up to us to want to do this. It's waiting for you. I think an enormous number of people get that clinical data warehouse together. 55 people on this list. I used to color code them by UC campus. Now I realize we are just one big campus with different zip codes. I have to thank a lot of collaborators over the past years at Stanford, UCSF, and NIH who got us a lot of our work done. I thank UCSF for endowment support, Priscilla Chen, Mark Zuckerberg for their gift. My lab has been blessed with 20 NIH grants from these 11 institutes of NIH. These 6 give me more, these 5 give me less, but I still love them. The governor's office invests in us I get to run precision medicine for the state of California with them. March Times, JDR, HMI, stem cell money, disease-specific foundations want us to work on their diseases. I always thank my admin and tech staff. I never get a grant or paper I have to go without them. Zach has been my friend and mentor for life. Sam and Keith for recruiting me to UCSF. I always thank my daughter, 15-year-old Kimmy. Why do I thank her here at this point? Because I want to tell all of you and really give this as the homework. We got to get biology and medicine more exciting to kids. Why is the first exposure to this dissecting frogs? Biology can be just as digital and cool, and this is what we got to keep them interested in, not just dissecting things. And I always thank my wife, Dongini Deshpande, molecular biologist, biochemist. She reads every major paper and grant that comes out of my lab, starts companies with me now, lets me go all over the world. Thoughts like this. Thank you very much. I went way over. I won't be offended if you run out of the room, but I think we have time for questions.
Yes, indeed. Well, amazing, inspirational. If you had a $10 million gift and your name was Shannon Zuckerberg, in whom would you invest? Here's an example. He's telling you there could be others sitting in this room.
Absolutely.
Who would like to ask a question? How many of you have IP filed among the posters up today?
3.
3 so far.
3 so far. Ah, yeah, great. Yeah, you've had a couple companies. There have been some good companies coming out here. I know. Yeah, I've heard of a few.
It's a rare chance, guys. Okay. Arvind.
First contestant. Go for it.
But there is also an adoption issue from the clinicians.
Yeah.
And somehow the clinicians need to change their mindset, DNA, whatever you might like to say, about how to let data inform their intuition and their subsequent choices.
Right.
How has that been for you, and how have you been able to work with them to make that transition happen in their mind?
That's great. So the real question to repeat is how are we going to get clinicians really thinking about this in the future? So first of all, you got to really work with really empowered and enabled physicians and clinicians, and you get them here on academic medical centers, right? That's why they're here. They could make much more money elsewhere. They want to be here because they're trying to still push the state of the art. That being said, there's certain clinical departments that are probably more advanced than others, so you pick and choose carefully. Diabetes, for example, they knew they had this kind of variation. They had no idea how to quantify it. We're thinking about how to run a clinical trial now. Not putting it into Epic yet, but just even a new poster. Can I just print out a new poster with our data showing which drug might work better or not? So you got to work with the clinicians that get it. That's true. But a lot of times people ask me, especially the basic biologists or basic computational folks, how do I even learn what the clinicians want, right? You are in a medical center here, right? Whether you know it or not. I send my computational folks to something really magical called grand rounds. Grand rounds is insanely early in the morning, but they usually feed you breakfast. And can you believe it? Every week, they get some world's expert in some disease to teach you everything they know. And the last 10 minutes of the talk is everything they want in that disease, right? We don't have a this, we're missing a this. And that's what they have weekly. Go to grand rounds. Maybe someday you're going to be sitting there and say, boy, I have data on this disease. Maybe I can do this, right? And there's your person to contact, right? So you got to go to these medical seminars and see what the unmet need is, because I will tell you, like I said on that third slide, that's the hard part now. What is the unmet need that's tractable given the data we have? I know I'm guilty of making everything seem easy. I admit that. I've been told that. I get it. But it's doable. It can be done. Question in the back there.
Yeah, hi, Liz Meliotis. So that diabetes data is very intriguing to me. So that's a lot of data and a lot of people on meds. So I'm curious, can you use genetics, and some of these are genetic targets, to essentially do a clinical trial in silico by sort of looking to see, you know, person has this genetic variant that affects this target and they went on this drug, but it didn't work or it did work. And that way you could bypass essentially doing the clinical trial.
Yeah, so in the end, it's about a clinical, so in the end, clinical trials are still mostly done to get drugs approved. And so they're already approved here at this point to do another clinical trial. So let me just tell you one truism I learned in 3 years now. Let me just tell you how ridiculous the state of the art is in medicine. Can you believe that after a drug is approved, in general, nobody bothers to study if the drugs work in their patients? So that's actually a truism, okay? It's approved, nobody wants to know now, right? The pharma industry's not gonna pay for that. NIH doesn't pay for that, right? So there's some exceptions, like some of big statin trials and stuff like that. But in general, the drug is approved, nobody even bothers to see if they actually work in real patients. So we're gonna change that. With or without the genomics, that data is incredible right now, because we can now start to see. I can start to make new metrics. You know, diabetes, the test we follow is the hemoglobin A1C, right? And we got hemoglobin A1Cs going up and down. We want them to come down. But a new metric now I can compute is improvement in hemoglobin A1C per dollar, right? Nobody is going to generate that. Nobody wants that number, if you know what I mean, right? But we can generate that number now because we have the claims data. We know the bills we sent for these as well. So I think even before genomics, it's incredibly valuable. Of course, we want to get to genomics. We're way behind, you know? I love Geisinger. I'm on their advisory board. I can see what Geisinger is doing, and Chop and Vanderbilt. We're aspirational to where they are right now. They would be able to do that kind of study in their sleep right now, looking at all this kind of patterns. We're just looking at more patients, but less. You know, someday we'll get there. Hopefully in the next 5 years, but we're not there yet. The question in the middle here.
All right, here.
We've spent a lot of time talking about the technical feasibility of a lot of these interventions and how to make decisions about treatment using this individualized information.
Yeah.
But to a great extent, the regulatory shuts that down and brings it to a screeching halt. Company that we work with in our lab had to do a new clinical trial in order to add one SNP to their panel.
Yeah.
What do you think is the road toward a world where regulatory is not shutting useful things down to such a great extent? How do we get more tractability?
So remember, so regulatory, what's that? Not in my state. Not my, if you know what I mean. So good question on regulation, right? So first of all, just be thankful we have a regulation. It's not state to state. Of course, New York does its own thing all the time, right? They have their own rules, but the rest of the 49 at least have some consistency there. But you're right. I mean, so in some ways, though, the future is going to be moving targets. And that's also why I try to get to simpler diagnostics, where a couple, you know, in general, ELISAs are a lot easier to get approved, and you know, protein tests and DNA, and RNA is even much harder. There's a cold chain and degradation, all that. So we could do better coming up with simpler tests, right? Reference lab versus LDT, those are all decisions you make early, and even which disease you go after, those are all strategic decisions that can, you know, get your company to float or sink right away if you choose the wrong way. Now, you're right, there are regulations out there, but I think there are certain aspects too, like clinical care, there's certain aspects that there are no regulations. Nobody tells doctors, there's no law that tells them which drug to use. The payer does that. The pharmacy benefits managers do that. And that to me seems really wrong. So that, it's a function of the insurance that really tells which part of the pachinko machine you go down with your ball, right? So I think in the diagnostic world, you're right. There's regulation. If you built the test and then all of a sudden want to change it, you know you're going to have to start a lot from that from scratch. Drug repositioning. It seems really ideal, but there are side effect problems there too that you're going to have to go get over. It's not. Might save a little bit of time, but you're still going to have to deal with that. So I think the regulations are there for a reason. But just think about it this way: that it is only paperwork in the end, right? If you can file paperwork faster, you get to these stories faster. So look, the way I think about life now. Look, you guys are here in Ann Arbor. You used to have big pharmaceutical companies here that pulled out a lot of. We used to have Roche in Palo Alto. They pulled out. Roche Nutley pulled out. Pfizer, where Viagra and sandwiches pulled out, right? There's a different world coming, right? Where those companies now are more interested in innovation through acquisition, okay? So what do? We can just create as many garage biotechs as we can, get them to some minimal level, and they're going to get acquired now, right? That's the game here, right? So you might not even have to do it all yourself, all the way to patients, right? You can go all the way, and you get amazing stories, but many times you're not even going to need to get that far. It could be for 24 months. All of a sudden, someone's going to, you can hand off the ball to someone else and take it further. So there's all sorts of models. David's got a question. Let's get to it.
Sorry. An NCBI, because
Of course.
it was really not obvious that this open data was in fact going to fly. PDB was closed. Russ Doolittle's sequence database was closed. Dave has the scars to show.
Absolutely. He and I were at the White House together getting the.
Jill and I watched it.
Yeah,
Remember that?
absolutely.
Yeah.
So I mean, I owe a lot.
It's great to see this.
That data is sitting there waiting for you. It's a tragedy if you don't use it. Now, I'll also make a little subtle shift here, right? I spent 80% of my talk talking about open data, and then I talked about UC data. Not that open. So there's still value in our private data sets. Someday we'll get a GenBank for clinical data, but that's a long way away because we're in a competitive spirit environment right now. We are trying to get patients from our neighbors, and we're not trustworthy yet to put clinical data out there. Someday, clinical trials data is as close as we can get. You had a question there, yeah.
So you're advocating.
All the microphones for the recording.
So you're advocating strongly for having scientists moving towards startups, right? But I think a lot of us are here because we're not really business type people, right? So is there like some sort of interface, intermediary that we can use that can do the businessy side of things?
Yes.
Again.
All right. So you have a CTSA program here. I'm sure they'd love to teach about entrepreneurship. They, like all the rest, have evening courses on faculty and students, entrepreneurship clubs, how to get started. You can also get other people to work on the companies with you, right? You might not want to quit your day job. The venture capitalists that are out there, investors, they have these things called entrepreneurs in residence. That's a fancy way to say unemployed, that they hang out and they're looking for their next jobs. They might take the ball and run with you. Those companies, they might change their idea radically because they're working full time with the company. We get to hang out in academia. Our next meal is paid for. They're going to earn it. So you got to have faith that companies might change the way things go, and that's okay. It's got to be okay. But I think what my wife taught me, 18 years ago, before we got married, what my wife taught me was just amazing. This concept that in today's world now, like it or not, you can criticize this, but like it or not, the very best business plans today look like the very best grants today, right? And today's exercise is how do you convince a room full of people to give you money to execute on your idea? It's not super different if they're in Bethesda or Sand Hill Road. And the one metric they're going to look for are, you got to change the world in 3 years. If you got an NIH study section, what do they drill in your head? Significance, significance, significance. You know, is this going to move the needle if they're successful? They keep saying that, just like Sand Hill Road. The whole point is, what are you going to do with this money? Now, of course, there's metrics that are different and there's deliverables that are different. But in my new worldview, I don't think about talking about companies as the dark side or the evil side. It is a continuum. My job as a faculty member now is to be a serial idea generator. I might have an idea someday that is so obviously going to help patients, I'm going to start a company on that. And lo and behold, medical schools, most medical schools in the United States, give faculty 20% time to go and do that. A lot of rules you got to follow. Strangely, they're the same rules on all of our campuses. You got to learn the rules. 20% time, I can go start a company. Boy, I might have an idea that really might help the world, but it's going to take a long time to chase that down. I'm going to write an NIH grant on that. And there's a continuum in between. And don't talk about it being the evil side or the dark side. You'll be amazed how many people can go back and forth now, because as long as people can see what you're doing in pharma or biotech or in your startups, you can see publications, you can go back. I've had many of my students do this now. But I think the world has changed. All I'm saying is that if you're going to come up with ideas, sometimes the best way to change the world might be through a company. And boy, most medical schools let you do this 20% of your time. So
And the best of the biotech and even pharma companies, especially the biotech companies, encourage you to publish and go to scientific meetings also, because they realize it gets better people, gets the work out there, gets bigger partnerships, earlier benefit. Atul, thank you so much.
Thank you,
Truly a
guys. Thank you,
memorable day.
everybody.